This training video guides you through the steps to successfully conduct a test using Neogen's Reveal Q+ Max for ochratoxin and the Accuscan Gold Reader. This presentation serves only as a visual guide to the written materials supplied with the test kit. To ensure accuracy in the performance of this test, please read and follow the test written instructions in their entirety. Each kit contains enough sets to test 25 samples. The kit includes test strips, Reveal sample cups, sample dilution cups, sample diluent, the QR code, quick response code, 1 bottle of high-level dilution buffer, and a box of 25 Max 1 aqueous extraction packets. To conduct the test, you also need the following: sample collection cups with lids, sample collection tubes with caps, filter syringes or Wattman No. 4 filter papers, dispensing pump or graduated cylinder, an agra-grind or bun grinder, a scale capable of weighing 5 to 50 grams, accurate to plus or minus 0.1 grams, a timer, a Reveal sample cup rack, a 100 microliter pipetter, 100 to 1000 microliter pipetter, pipetter tips, distilled or deionized water, Max 1 aqueous extraction packets, and the Reveal Accuscan Gold Reader. Remember to always store test strips in the stay-dry tube at room temperature. Do not freeze the test strips. Treat all used liquids and labware as if contaminated with ochratoxin. Neogen recommends gloves and other protective apparel, although they're not required during testing. Be sure to follow your company's policies. Avoid cross-contamination by using clean extraction supplies for each sample. Neogen's Accuscan Gold system consists of a lateral flow reader and data management software that interprets Neogen's Reveal Q+ Max test strips for mycotoxins. The system provides an easy method to objectively read, store, and analyze results. The Accuscan Gold Reader must be charged before use. Charging takes approximately 4 hours. The reader can be charged by attaching the power cord and the USB cable directly to the reader, or attaching the cord and the cable to the cradle and inserting the reader into the cradle. Once charged, the reader's power should last up to 3 days of regular use and up to 2 weeks with no use. To turn the reader on, push the button on the left-hand side below the screen. The main menu screen is used to access all sample testing, sample results, user identification, and other test support utilities. Prior to running a sample, you must enter the lot-specific QR code. To do this, place the disposable QR code contained in the kit into the QR code cartridge. Select the Scan QR code icon from the home screen and place the QR code cartridge in the reader. The specific information is automatically downloaded into the reader. Alternatively, you can manually enter the QR's 39-digit code. For instructions on how to do this, see the Accuscan Gold User Manual. It is important to scan the QR code for each new kit lot. For this example, we will be using the wheat protocol that uses curve 1. Please consult the kit insert for commodities that use curve 2. To prepare the sample, begin by gathering all of the materials required for the test. The sample to be tested should be collected according to accepted sampling techniques. See the GIPSA sampling protocol or contact your Neogen representative. Obtain a representative sample by grinding a minimum of 100 grams of sample so that at least 95% of the ground material passes through a 20 mesh sieve. The particle size should be about the same as espresso coffee. The sample is extracted with a ratio of 1 part sample to 5 parts distilled or deionized water. For this example, we'll use 10 grams of ground wheat with 50 milliliters of distilled water. The USDA GIPSA approved method uses 50 grams with 250 milliliters of distilled or deionized water. Weigh out 10 grams of ground wheat into a sample cup. Pour the contents of 1 Max 1 packet on top of the weighed sample. Make sure the entire packet is emptied into the sample cup. Add 50 milliliters of distilled water to the sample cup. Vigorously shake by hand or mechanical means for 3 minutes. Allow the sample to settle and filter with a filter syringe or Whatman No. 4 filter paper. Alternatively, you can pipette 1 ml of the sample into a 1.5 ml microcentrifuge tube. Centrifuge for 1 minute using a microcentrifuge. For each sample you're testing, place sample collection tubes and reveal sample cups in a sample cup rack. Add 100 microliters of sample diluent into each red sample dilution cup. Then add 200 microliters of the sample extract. Mix up and down with a pipette at least 5 times. Transfer 100 microliters diluted extract to a reveal sample cup. Use a new pipette tip for each sample. It is important to make sure the pipette tip is fully charged with liquid and doesn't have any air bubbles prior to dispensing the liquid into the cup. Place a Reveal Q+ Max for Охратоксин test strip into each sample cup with the sample end down. Allow the strip to develop in the sample cup for 5 minutes. During this time, prepare the Accuscan Gold reader to read your results when the incubation is complete. To do this, select the test category Mycotoxin Q+ Max using the category drop-down menu. Select the Q+M Ochra Curve 1 test type using the test name drop-down menu. Next, select the appropriate username under the username drop-down menu. Select Run Test once the test information is selected. A window will appear asking the user to input a sample ID. Enter the ID and press OK. If you do not want to add the sample ID, simply press OK while leaving the field blank. Following the 5-minute incubation, remove the strip and insert it into the R-labeled strip cartridge. Fully insert the cartridge containing the strip into the reader upside down, sample port end first. When correctly inserting the cartridge, the test line faces downward into the reader. The reader's green light glows when a cartridge is inserted. The reader automatically begins analyzing the cartridge. Within 2 to 4 seconds, the green light turns off and 1 beep indicates the completion of the analysis. Removing the cartridge prior to completion can result in invalid readings. It's important to note that readings must be conducted within 1 minute of the completion of the 5-minute incubation. Readings conducted after 6 minutes may be inaccurate due to overdevelopment of the device. A results screen appears. Images of the test and control line zone appear on the center of the screen. The sample ID and result appear below along with the line intensities. To see the full test image, select View Image at the bottom of the screen. The test results are automatically saved. To reread the same strip, select Retest. To test a new sample, remove the cartridge and select New Sample ID. Input the new sample name and insert the new test strip in the cartridge into the reader. The test category, type, and user remain the same as the previous test unless the user returns to the main screen to edit these fields. To transfer the results to the Accuscan Gold Data Manager program, insert the reader into the cradle or attach the USB cable to the reader and attach it to a computer. Open the Accuscan Gold Data Manager software on the computer. Select the sync icon on the software to pull data from the reader to the computer. Any samples that show greater than 20 ppb on the Accuscan reader will need to be diluted and rerun. Dilute the filtered sample 1 to 5 with the yellow labeled dilution buffer using a sample collection tube. For this example, we will be using 100 microliters of sample and diluting with 400 microliters of dilution buffer. Mix by pipetting up and down at least 5 times. Add 100 microliters of sample diluent into the red sample dilution cup. Then add 200 microliters of the diluted sample extract. Mix up and down with the pipette at least 5 times. Transfer 100 microliters diluted extract to a Reveal sample cup. Place a Reveal Q+ Max for Охратоксин test strip into each sample cup with the sample end down. Allow the strip to develop in the sample cup for 5 minutes. Following the 5-minute incubation, remove the strip and insert it into the R-labeled strip cartridge. Fully insert the cartridge containing the strip into the reader upside down, sample port end first. You need to multiply the result displayed by 5 to account for your dilution. If you have any questions regarding the use of the AccuScan Gold reader, please contact Neogen. Manuals are also available. Please contact Neogen for more details on our current and future Q+ Max test kits.